you mentioned some really important things, which is the lack of reproducibility of research. And it's something we should all care about because we care about having medicines. And right now, the failure rate for drugs is astronomical. You have a better chance at roulette by far. So when you look at the causes of it, about 50% are poor preclinical research that's not reproducible. And that came from a study at Amgen, where 80% of the about 60 studies, they were not able to reproduce. And they had 100 scientists for 10 years. So I got interested in this area because I have 11 companies I've started. 5 have multi-billion-dollar exits. I said 4, but I forgot about one. And one of them was a company where we automated cytology labs, which also, like cell culture, has been ignored. So I look for pockets where nobody else is, and where the solution crosses fields. So we're crossing software and life sciences. So we started Thrive about 7 years ago, and as I mentioned, it's my 11th company. My first 3 were software companies, all AI companies. One sold to Oracle, one sold to Fair Isaac, each for about $1 billion, and then moved into life sciences. And by the way, even though I have maybe the most successful standalone AI company in terms of returns, we never talked about it. So I'm a little bit suspicious when people do that a lot. So Marty, I did have some slides.
Yeah, I'll start it right now.
Sure. So I don't like introducing myself without introducing my co-founder. So my co-founder is from Caltech and is responsible for developing the sequencing instruments that Illumina sells, which is a multi-billion-dollar product, and has enabled genomics to be what it is today. And he did that for 25 years, and he wants to duplicate that with live cell biology, which is really short on data. So we're automating live cell imaging with analytics for drug discovery, basically. Next slide. So I wanted to just go through key points. I always have this this fear that it happened to me once, that a fire alarm goes off and I only have the first few minutes. So I want you all to understand what we're doing. So the instruments that we have do not require FDA approval. We have 15 users that are contract research organizations and pharma. We have collaborations with the top organizations: Harvard Stem Cell Institute, University of Texas Medical Branch for infectious disease, the Broad Institute for tissue culture, Center for Genomics Medicine, at Massachusetts General Hospital for single cell tracking. We like developing our products with customers, and that's what we do. So I mentioned the team. There are 19 employees, and our chairman was president of Thermo Fisher, the largest player in our space, and Brock Reeve is executive director of Harvard Stem Cell Institute. There's been very limited innovation in this area. Just like Cytek found the cytology labs where we automated pap smears, and pap smears went from being every year to every 3 years because of the accuracy of our instruments, and it was really all about collecting data. This area is also ignored, and so we've filed 78 patent applications, 25 have been issued. We basically, somebody's going to have to buy our products or buy us. We've raised $30 million to date, and actually mostly from family offices. Our largest investor is the Estee Lauder family. They invested in 2 of my previous companies, AI company and Cytek. We also have 7 angel groups and a lot of industry folks like former CEOs of Novartis. So we're raising money. We're finishing up a $5 million convertible note, converts into a Series B at a discount. I would very much like to finish this up in a couple of weeks, so I am very interested in investors if they're interested in participating. And we expect the Series B to be in the fall. We already have a $10 million term sheet, but we want a slightly different group that can really add a lot of value. But I'm also, I have a letter of intent for an acquisition of a company very similar to, in the space as us. They're the leaders in environmental control of cell culture. They'll do about $6 million in revenues. They're profitable. I'd like to acquire them. I'm using a debt with warrants. I need $5 million in cash. I have $3 million committed. So if somebody's interested in debt-like structure, also interested in that. So next slide, please. So I'd say most industries have kind of dirty secrets that you don't know about unless you're really involved. And cell culture is one of the biggest in life sciences. It is done manually. You take the cells out of the incubator in plates, put them under the microscope. A researcher spends on average 2.5 hours a day looking at the plates, and they put the plate back into the incubator, and guess what? There's no data that's retained. Maybe 1% of the labs are trying to retain data, and it's spotty. So it leads to all kinds of things: genomic drift, cell stress, and mislabeling. Mislabeling rates are absolutely extraordinary, 11% to 30%. Next slide, please. So it's really summed up well by one of our customers, chairman of the biochemistry department at US Medical School, who's been using our instrument for about a year. Current tools are failing us because they hurt the cells, only give us 2D, single point in time images, and they don't capture contextual data or close to enough data. And we're really solving all of those problems. But currently, the instruments that are available are very far removed from the biology. You can't follow processes over time. Biology is three-dimensional, but most of it's 99% of the instruments and images are taken at one focal plane. Next slide. So we already talked a little bit about the lack of reproducibility in Amgen and many other studies. And basically, data is not comparable because it's done manually. It's more of an art than a science, and we're turning it into a science. Next slide, please. And we're doing that with instruments with a very heavy content of software. We're automating the imaging process so that you get the same set of images every time. We provide really for the first time analytics. Documentation is extremely important. We capture everything. And our goal is to have reproducible data for cell culture, stem cell culture.
Tom, did Amgen give a reason why they couldn't replicate 90% of their?
Yes, so 50% of it related to poor cell culture.
Okay.
Mislabeled reagents, cell stress, not cherry picking of data. So they see something interesting and, or say biased data, as opposed to having a record of all the images. And then the other 50% is from kind of clinical poor animal models. Like, as many people know, we've cured every disease in mice and far fewer in humans. So that same person who said all the problems that we have, 2D, single point in time, used our instrument and after about half a year said, Thrive will change the world. Thrive allows us to understand and see living dynamic cells and tissues in 3D over time with remarkable images. And really, we're really providing for the first time data sets that have never been available before so that you can do data mining, machine learning, good documentation, and breakthrough insights, which we've already seen among our customers. Next slide, please. So just to talk a little bit about the products, for a company at our stage, we're just beginning to sell products. We have two products, a benchtop single plate imager and a automated 50 plate imager. We'll be bringing out a series of software modules to be sold by subscription starting at the end of this year that really address key workflows. Early next year, we'll be bringing out a less expensive instrument, and then in a few years, increased automation. Next slide. So
by software modules, you mean software modules that work with your hardware only?
That's right. That's right. So in the future, we would like, our objective is to be on every microscope because we're really like a 4th generation microscope, but the imaging that is taken by microscopes just isn't sufficient. It doesn't take all of the cells in all of the wells, doesn't take it with different focal planes. So for now, we have to use ours, but we view ourselves as a software company, Marty. So if somebody else came along and did it, we'd be glad, but I don't think anybody can, but not without violating our patents, and it's very difficult. So I just want to mention, this is our 50 plate imager where you can remotely image plates on whatever schedule you want. Those data points there are every 4 hours, went on for months. It was around Christmas and New Year's. And our sites are generating 2 to 3 terabytes a week, which, because my co-founder comes out of genomics, we're able to handle very well. Users don't even know it. The next slide, please.
There's a question from the audience. Where are you in terms of cash flow, EBITDA?
So we're just starting to sell our product. We just hired a commercial team. We expect to do about 4 million without the acquisition in the next 12 months. We have a burn rate of about $300,000 a month. We expect to get to break even in about a year.
And we're gonna get a non-confidential pitch deck if we want to
Sure,
share it with
sure.
everybody.
Yeah.
And you're looking at 5 million by when, and what's the minimum?
So I have 4 million of the 5 million on the convertible note.
Okay.
And so I'd like to finish that up in the next 2-3 weeks. I told Marty I would make a special arrangement. Usually it's $250,000 minimum, but we'll look at a smaller amount, aggregate investors from IvyFON from these presentations. I have about 500,000 from 750,000 from people I've met through here. So it depends on who can the investor add some
Is
value.
it Tribe Capital? Is that's when we're getting the name mixed up? Thrive Bioscience.
There's a Tribe Capital also,
Capital,
right?
yeah.
And Tom, just another question. Have you gone through, I mean, there's more than one Lauder family office, right? There's like 5 or 10 of them, right? Ronald Lauder has one. The grandson has one he's out in Silicon Valley.
We're in 2, one in New York and one in Palo Alto.
So, right, so that's the grandson, right?
Which is, the grandson, Gary, is really more a tech investor.
Yep.
And when I first set up a breakfast with him to talk about Thrive, he said, "Tom, I always love seeing you, and you made a lot of money for us, but we're not going to invest in this because we don't really invest in life sciences." But by the end of the breakfast, he said, "I realize now it's all about the data."
It sure is, right? It's about the data. And it's really funny because I have a master's in AI, and what we studied was AI visual systems and this kind of way of using fuzzy logic to define what the data is, which is what you're using that application right now.
Yeah.
So
Absolutely. So this is maybe the most important slide, which is really our imaging, which is kind of our secret sauce. So for the first time, we captured thousands of 5 megapixel images. We do it about every 250 milliseconds. Nobody is within factor of 10 from us. And we are able to image at 100 focal planes, 2 to 50 micrometers or microns apart, selected by the user. A typical scan in minutes generates 1.5 to 90 gigabytes of data in 10-15 minutes. This has just never really been available before. And we're able to do something no one else was able to do, which is to track a single cell over time, so you can actually follow it. It's very hard to even find it because it's just one little cell sitting in this giant well. It's a needle in a haystack computing problem. Next slide, please. Just wanted to mention quickly documentation. It's incredibly important. It's one of the causes of lack of reproducibility. We make it extremely easy. You can put barcodes on the incubator that the plates come from, the lab notebook, the reagents, and one of my patents is in basically the Internet of Things, which is to connect and collect everything that's going on in the cell culture lab, which is particularly important as we move into cell therapeutics and you're putting these cells back into people. Next slide, please. So competition is limited. As I mentioned, it's mostly digital microscopes. They don't produce the kind of quantities of data. They typically kind of biased because somebody sees some interesting images and we'll click an image of them. Most have trouble focusing, particularly when there's debris, which often happens when you're studying biology, and our algorithms avoid that. And we made it very easy to fit in a workflow. We assume that the person knows nothing about engineering, and because biologists typically aren't instrument people. Next slide, please. So I just wanted to show a little bit of data. I mentioned that 11 to 30% of all cell culture cell lines are mislabeled, which is a accumulation of manual processes over many years. That comes from the FDA and NIH. And for the first time, you're able to easily see by just looking at things that are different than all the others. So this is a chart of cell growth from the Broad Institute, and they noticed that there were some cells growing faster than the others. We asked them about it because we thought, oh, maybe they're trying to optimize their cell culture, which is a big application, but no, they were mislabeled and never would have found that.
I have another question. So just to understand where mistakes are being made today, do the current researchers use like probabilistic models or statistical models to like estimate where the cell culture is? Is that what's going on, or
there's nothing, Marty?
they're not even there?
It's just manual human eye, right?
I know a lot of people in the medical profession, and they're not good statisticians. So
no, they're just not. It's really an art, not a science. And one of the things that helped us at Cytic, so Cytic was sold for $6 billion to Hologic, and part of the reason for the valuation was that we had the cells from the pap smear sample, and then we're able to run, so all the HPV tests are run on Cytic instruments. And here we have the cells. We have all of the, by being close to the cells, we literally are creating these huge databases. Next slide. So I want to just quickly go through some images. This is something that never had been able to be done before. This is for COVID-19 research. The researcher who said that Thrive will change the world was able to look at viral plaques, which are holes in cells from a virus. It was used for COVID-19. And currently you have to stain in order to count them. So you have to kill the cells. They didn't want to stain it because they don't want to touch the virus again. And they were easily, with our instrument for the first time, didn't have to stain. But more importantly, they could follow the progression of the plaque. And there's actually a lot of data in how those plaques or those holes form. Next slide. So this is something that also wasn't able to be done in wound healing. This is a scratch assay, and we basically measured the scratch, which is the wound being filled in by cells, and we did it quantitatively. So for the first time, the researcher was able to compare over 100 different combinations of drugs to see which one was best, just not available previously. Next slide, please. So this is tracking a single cell. This was from Harvard Stem Cell Institute. The importance of this is that CRISPR or cell engineering, you end up with one cell, and then you have to grow it. And we got a call from Mass General Hospital saying, we have to wait 2 weeks until we can even find whether the cell that we've engineered is alive. They have to wait till it's a clump. But we can actually track a single cell. In this case, it became 2 colonies. Next slide. So one of the fastest growing areas of cell culture is tissue culture or organoids, because it's a better model than a bunch of cells just flat on the bottom. It actually, you can simulate an organ, and it falls between cell culture the way it is at the bottom, some cells at the bottom, to between that and animals. And there's a huge need for something that's better at all stages. So with our stacks, we're able to image organoids unlike anyone else, and you can actually see the differences as you go up and down the stack. Next slide, please. So this cell culture, amazingly, is a $29 billion market. We're selling directly. It is a sale to researchers and somewhat technical. And we believe that we're going to have 150 million in revenue in 5 years. I've done that before. Basically because we're applicable to every segment of life sciences. We did not know we were applicable to infectious disease, but when COVID-19 happened, obviously everybody tried to figure out how they could make their product useful, and if it was useful. And we were astounded at what customers were able to do. So we're also going to add more and more products, and our objective is to become really the data provider in cell biology.
So at that 150 million, how much is that as hardware versus software versus data?
So it's a little bit hard because we do bundle, but we are planning on unbundling, and to a certain extent, they would not buy the hardware if they didn't have the software. So you could say it's all because of the software, but if you kind of make an estimate of unbundling, I think we end up in 5 years at about 50% is from software. About 10% is from consumables because we're adding some automation, some fluidics, and that rises very, very quickly.
Who owns the data?
Great question, Marty. So one of our investors is the founder of Viva Systems, which is the largest clinical trial software, fantastic company, public company. And they're the leaders in clinical software. We want to be the leaders in preclinical software. And they were able to retain ownership to the data. Excuse me, not ownership, but a license to the data, and we're going to follow the same.
At what level of granularity?
So we're still experimenting with that. So nobody has had any problem with us having the data internally as long as we aggregate it and it's not identifiable, up to the point where they start adding proprietary drugs. So we can work well with that, and we have found academics in return for a break on the instrument are glad to have it identifiable and to supply the data through the entire research.
Right, so there's multiple cases of just data companies who collected oncological data of a specific ethnicity, let's say, and they were able to sell that data for billions of dollars.
So Marty, that's a really good point. So for patient-derived cell lines, there's the opportunity, you mentioned racial race in terms of there are different responses in healthcare. And it's a priority of the FDA. You have to show them that it doesn't just work for one group, it works for all. But there's nobody who collects where the cell lines came from.
Right.
And so we see that as actually a really big opportunity in really more 2 to 3 years, where there's just a whole nother level of data that goes with cell culture. So you could actually model at a much earlier stage whether there are any genetic differences that might occur.
So that could pivot into huge opportunities
In
like we're
clinical,
seeing.
yeah, in the clinical area and cell therapy.
I mean, based on what you have just right here, just in this presentation, I assume there'd be a lot of interest from strategics, J&J. I mean, I work for Medtronic.
Yeah.
I would imagine
So-
that. it's not,
So
Regeneron I mentioned.
so interestingly, there is not that much interest compared to what you see in pharma.
Okay.
And the reason is there are about 14 or 15 companies who would be likely acquirers. And they're all basically optical or microscopy, but mostly research tools.
Okay.
So if you look at Thermo, PerkinElmer, Agilent, Sartorius, Stedham, these are all likely, very likely acquirers. They do hardly any, if no minority investments. And we know them all through advisory board, et cetera, and they
Sure,
basically say, we acquire.
Yeah.
I don't know why they
Don't
haven't done
invest,
that.
Yeah. We see a lot of, the collaboration agreements where put in initial money, then you meet certain milestones, then they acquire.
Absolutely.
Yeah.
And I ran a pharma, public specialty pharmaceutical company that's everyday activity,
Sure,
right?
sure.
Is licensing in, licensing out.
Yeah.
Just, it doesn't happen
Well-
in research tools, which is one of the reasons that family offices have actually bridged,
Great.
bridged a lot of that.
Yeah.
We have about 5 minutes left.
Okay, the next slide. So I just, we can actually skip that and go to the next couple of slides. We kind of talked about this. Next one. So just want to talk about M&A in line with what we were just talking about. There, the multiples in research tools are actually number 2 to biotech. They're really quite extraordinary. In M&A, there's limited data, but there's average sales in M&A are 10 times trailing revenues. And that includes a lot of companies that were hurting because that's why they're being sold. If you look at public companies, it's about 35 times trailing 12 month sales. And we've been approached by at least half of the ones there and several times by several of those there. So just to kind of summarize the next slide, We want to transform this field of starts with cell culture, but cell culture is the basis of cell-based experiments, which is the basis of most biology. We're basically doing for this industry what sequencing did for genomics. We view ourselves as an Illumina type company. Almost all of our customers are conducting drug discovery, and each startup is just way too hard to not have an aspect of it of changing the world. And so we would really like to improve drug discovery. We all have family members or friends who, so that's what motivates me when you're in an airport and your flight is canceled, which just happened, and you have to sleep on the airport, things like that. Might as well try to really have a big impact. So just a few quotes from customers, because that's far more important than me telling you anything. Is one is remarkable resolution. We see things we have never been able to see before. Number two, second one, tracking single cells and colonies in 3D is game changing for drug discovery. This is what cell culture and cell imaging should look like. So thank you. And do we have time for?
Did you apply for any grants from the federal government, NIH or so on, or just too long of a process?
We have not. I'm a commercial oriented person, and I think we could benefit.
Yeah, it's non-dilutive money, as you know.
Right, but what we have done is some of our customers we've supported them,
Okay.
and then they buy our instruments.
Yeah, that's nice.
But I'm looking for somebody who can help us on that, because I think there's some opportunities. It is a long process, but say it's 6 to 9 months, but then 6 to 9 months later, you look back at it and say, I could have had I could have had non-dilutive financing.
The client got $8 million in his research, of course, different areas, but money's there.
Yeah, it is.
Same with DOD.
Yeah, DARPA.
Yeah.
DARPA is absolutely incredible, and they've elevated pandemic life science research.
Plus just another validation of one of the cycle.
No, you're right. It's an area I haven't done as much as I could.
Tom, if an exit is strategic, at what point how far do you grow the revenues?
When's the optimal time?
Yeah,
So, that,
by the month.
well, I think it's probably a couple years away, and I think some of the key criteria are certainly an install base of, say, 50 instruments. Papers take a long time. Peer-reviewed journal papers. We have several that are in the works by researchers, but it's a slow process. But that's an important validation. Having grants, is another.
Get the patent application on file before you publish the paper. So.
Yeah, so we're pretty good about we have a full-time patent counsel who works for minimum wage and then equity. Otherwise, we couldn't afford it. Couldn't afford that. So I think it's really validation from the academic community with papers, customer base, and then people having extraordinary results, which we already have. But I think pharma is shy because of the reproducibility problem, so they're going to want to see more. But I think that could all happen in 2 years. You can never predict, and we're not trying to sell the company. We're trying to build value with an eye towards selling it. But it's a mistake to count on a flip or anything like that.
Build it first.
Build it first, but we think about what they would want.